What up, botanists? So now we're going to look at, in a bit of detail, how we could inhibit germination and how we can promote it. And in fact, the promoting part, get your skates on, you're going to do the work. So let's have a look. By the end of this, you should be able to explain how one can both inhibit and promote the germination in plants or in seeds, and probably why you would want to do it as well. So you want to remember that the germination is the process of a seed sprouting. That's pretty straightforward. So inhibition of germination. Basically, they're often treated to inhibit them, which means to slow down or stop their growth. Why would we do this though? Well, one, it allows us to save seeds for the following year. Two, it allows us to maintain seeds for a seed bank. And this is particularly with endangered species or when we want to maintain a bit of biodiversity. These are seed banks, and you'll notice one thing with the way both these people are dressed. Essentially, seeds require optimum conditions to germinate. All right. Therefore, these are what we alter to, inhibit their growth. So if we want to inhibit their growth, we take the conditions out of the optimum. All right, so how do we do this? There's three ways we're going to look at straight away. First is cold storage, and if we go back here, we can see that everyone's wearing nice big fluffy jackets. Cold storage. Basically, refrigeration or freezing for long periods of time reduces germination. It just slows it down. Now, the way this works is it inhibits the activity of enzymes. Those enzymes really need to be active at certain temperatures. And these are required for cell respiration. Without this, cells become dormant. They don't die, or hopefully they don't die, they just stop. Now, there are negatives. See, there's a minus sign with ifs, so negatives. There's high energy cost associated with refrigeration and freezing. And unfortunately, seeds may fail to germinate when they're finally exposed to the correct temperature. So when we finally get ready to plant them, we warm them up, they still may fail to germinate. Now, this is still probably the primary way this is done, even though it's high energy costs and the seeds may fail to do it. It's the primary way to do it because it's the easiest. For example, vacuum packing. Vacuum packing removes oxygen supply for respiration, so the seeds are stored without any O2 and under pressure. What you can actually do is, instead of vacuum packing, you can displace all of the air with nitrogen, so there's still no oxygen, but it's full of air and it's not quite under the pressure. This is massively labor intensive. Like, this takes a long time and it's harder to do than, say, cold storage. Desiccation and dehydration, well, basically this just removes the water, and this stops the swelling and rupturing of the seed coat. So if we keep them nice and dry, they won't germinate. Now, desiccation is what happens naturally when we just leave it out in the sun and that stuff. Dehydration is the one we do artificially. So there are two different processes there. And we also want to promote germination. So once we've got those seeds, we then want to find ways to make them grow forward a bit earlier. There are several ways to do this. They're a bit more straightforward than the three ways we've got of inhibiting it. So I'm going to let you guys summarize it. And it's just page 79 to 80 of your textbook. Now, I want you to write down what they are, how do they work, and what plants are they for? Are they for something specific? For example, lime pelleting, is for a specific type of plants, or is it a generalized one which works across the board? So they are heat, beds, lime pelleting, and misting. All right, that's it, and I'll see you in class.